Professor Dr. Chitrita Chatterjee is a professor of the Department of Microbiology, again from this prestigious institute, IPGMER and Cecil Hospital. She has done a graduation from NRS Medical College and MD in microbiology. She was a gold medalist, again from this institute. Started her career as a second medical officer in primary health center and then promoted to block medical officer of health catering more than 100 villages. Encountered the multi-drug resistance outbreak in her time at BMOH, associated with Medical Education Service in 2012, ex-head of the Department of Bardwan Medical College and NRS Medical College, authored in multiple publications which notably includes topics of arboviruses and Japanese encephalitis virus, rendered quite a decorated research on neonatal gut flora since her days in NRS Kolkata, guided multiple MD thesis projects and acted as a co-guide in a PhD thesis, honorable author of a book on practical microbiology, which is about to get published. So ma'am, stage is all yours. You can present your session.
Good afternoon to everyone. I'm really indebted to this organization as they have allowed me to address this honored. Hello. I'm indebted to the organization as they have allowed me to address this August gathering. So I am typically a microbiology teacher, a professor of microbiology, so I will be talking only about microbiology to you all. So my topic is methods of replacement of gut microbiome. But what is the gut microbiome? The gut microbiota is the aggregate of all the gut flora, including bacteria, archaea, fungi, viruses that live in the digestive tracts of animals. The gastrointestinal metagenome is the aggregate of all genomes of the gut microbiota. And the gut is the main location of microbiota in the humans. So before that, I want to tell you a bit more. That is, whatever we do in our labs is concerned mostly with aerobic bacteria and very few anaerobic bacteria. But in case of gut microbiota, the situation is just the opposite. Everything is anaerobic microbiota, and there is very little aerobic bacteria involved, which is actually not aerobic, but aerobic and facultatively anaerobic bacteria. So that is what we get. So most of what lives in our gut remains unknown to us. Not only to us in India, but to the rest of the world. The problem with the gut is that most of the world, the developed countries, they have started research on the microbiome of the gut, but they have forgotten the culture on the plate culture that we do in our lab, which is usually abused as something which Robert Koch had done, and they just look down upon it. But right now, the need of the hour is culturing the bacteria, because we are not only concerned about the bacteria in our gut, but also the bacteriophages that remain in our gut. Until and unless we culture the bacteria of our gut, we will never be able to get hold of the bacteriophages, which have been only found out by molecular methods, and their presence is there only in the computer. So their actual presence has not been there in the lab, and we know very little about them. Even 5 years back, most of the people who were working on human microbiome, they considered that the most of the gut biota was not cultivable. But right now, in the last few years, there has been a very big progress. And what has the progress told us? It has told us that most of them are cultivable. So a huge group of bacteria that were just labeled as non-cultivable bacteria, non-cultivable but viable organisms living in the gut, have been designated as bacteria that can be cultivated. And this is a very recent publication, a publication in Nature, which was there only in the last month. So things are really happening regarding human microbiome. So let us go on. So what does it do? What does the microbiota do? It has effects on colonization, resistant to pathogens, maintains the intestinal epithelium, maintains the intestinal mucous membrane, metabolizing dietary and pharmaceutical compounds, controls the immune functions, controls behavior through gut-brain axis. All of my previous speakers, the eminent speakers who have spoken on this stage, had something to do with my topic. I will come to that why, because each one has said things that really involved the gut microbiota. Now, what is the composition of gut microbiota? The microbial composition of the gut varies across regions of the digestive tract. The colon contains the highest microbial density, about representing between 300 and 1,000 different species. Bacteria are the largest to the date, best studied component, and 99% of the gut bacteria come from about 30 to 40 species. Up to 60% of the dry mass of the feces is bacteria. Over 99% of bacteria in the gut are anaerobes. But the cecum, here there is a typo error. The anaerobic bacteria reach the highest density. In the cecum, the anaerobic bacteria have the highest density. It is estimated that the human gut microbiota have around 100 times as many genes as there are in the human genome. So now just imagine there are so many genes, a huge metagenome sitting in your gut. And do you think they have got no control on you? Don't be so sure. So you are what you eat. So it comes back to the old proverb again. So that microbiota dictates your nature, your depression, your anxiety, and a lot of factors. Actually, the topic is so large, maybe I won't be able to say much here, but still I will try to focus on a few points. Now here we see the intestinal microflora. We know that the stomach is very acidic. It has the minimum number of microflora and maximum is in the cecum. And then we have the colon and the appendix with the largest number of bacteria, 10 to the power 9 to 12. According to some sources, it is up to 10 to the power 13. Now the gut microbiome. In humans, the gut microbiota has the highest numbers. The approximate number is 10 to the power 13 to 10 to the power 14. In humans, the gut flora is established at birth. We had been studying neonatal gut flora in NRS. We have presented our papers in several places, and we want to continue with the study even here because it's a very, very interesting topic. In humans, the gut flora is established at birth and gradually transits towards resembling the adults by the age of 2. At the age of 2, the intestinal epithelium is matured and so is the intestinal mucosal barrier. Both the intestinal mucosal barrier and the epithelium, they have to do a lot with the bacteria that resides there, which we were falsely calling commensals all these days. I'll come to that. It's not a commensal, but it is highly interactive. This barrier is essential for supporting a symbiotic relationship with the gut flora while providing protection against pathogenic organisms. Now comes the relationship. I was talking about the relationships. It is here. It is not merely commensal, a non-harmful existence, but rather a mutualistic relationship. Some human gut microorganisms benefit the host by fermenting dietary fibers into short-chain fatty acids such as acetic acid, butyric acid, and also by metabolizing their end products are GABA and serotonin in some cases, and a lot of other substances are there, which I will not be able to say in such a short time, but we all know. It is a very old question where we say that the gut behaves like an endocrine organ. My previous speakers have also talked about it. So the gut behaves like an endocrine organ. And next we come to intestinal bacteria also play a role in synthesizing certain vitamins. My previous speakers have spoken about this also, vitamin K, metabolizing bile acids, sterols, xenobiotics. Now what are xenobiotics? Do we know about xenobiotics? Something very, very harmful to us. And the easiest example is microplastics that we are taking in with our food all the time without our knowledge. The systemic importance of short-chain fatty acids and other compounds is that they are hormone-like in function. Now, dysregulation of the gut flora has been correlated with a host of inflammatory and autoimmune conditions. So let's come to dysbiosis. The gut microbiome can be dysregulated, or there may be dysbiosis due to a variety of factors, due to antibiotic use. This is most important. Rather, I should say antibiotic abuse. And not only that, we are very careful about, I think there is a session on this topic about monitoring of that is antibiotic stewardship of the institutions that should be carefully adhered to, and we know this has been causing more harm than good. So here, other than antibiotic use, I will talk about one more thing here that is not written in my presentation, that is about use of immunotherapy. We all know nowadays that immunotherapy has become very, very important. And I have lost a very dear person to me, my husband, because of this, because I keep on talking about this, because he was given immunotherapy and the result was gut dysbiogenesis, and that ended in gastric bleeding. So that was the end of the huge effort that everyone had put in for saving him. So anyway, I should not go on with it. Poor diet, we all know about it, we have been talking about it. Chronic stress. Now, poor diet does not mean that I'm talking to dietitians, I know that very well, that poor diet does not mean that there is no intake of food. Rather here, poor diet means there is low intake of fiber in diet. So what happens is exactly, you get used to a high fat diet, okay, a high fat and a high carbohydrate diet. I know all of you know about it. It's very good to eat, and as dietitians, definitely you won't prescribe things like that to your patients. But, there's a but we love to eat them. And then what happens? We keep on eating them. And what happens? The gut microflora that used to live on those fibers, dietary fibers, gradually fade out, and they give way to the microbes that feed on the sugars and the fats. So in this way, we are killing the healthy microflora by an unhealthy diet. And then comes chronic stress, infections, chronic diseases, diseases, environmental factors, medications, especially proton pump inhibitors, mode of birth. We had made a study in NRS on this topic, that cesarean section babies, though it is known that even they have some gut bacteria, but still the number of gut bacteria from the mother was less in cesarean babies than in babies who were born by natural method, naturally. Well, so now we come to a very important part, that is dysbiosis in ICUs. So this is something very, very important. So here you have antibiotic use again, use of and critical illnesses, nutritional illnesses, gastrointestinal transit changes, environmental factors, sepsis, and last but not the least, I'll repeat again, that is immunotherapy. Whenever the patient gets immunotherapy, be very, very careful about the gut microbes. That is the first thing to be harmed. So that has a tremendous impact on the gut microbes. And the patient may have gastric bleeding or even gastric perforation. So look after the trauma. Immunotherapy is not something very good, though it's a life savior at times. So what are the remedial measures? Now let's come to our topic. So first thing, let's begin it easy. All of us know as dietitians that it will be dietary changes. So modify the diet. It promotes a diverse and healthy gut microbiome. Here we are thinking in terms of the microbiome, not in terms of the patient's health. Whenever we think of the gut, we think of it as an empty tube. It's not an empty tube. It's full of life, it's full of genes, and it's full of interactions. Remember that. So dietary modification can bring back your gut microflora. Now comes the 7-day gut reset. I think all of you know about it. So what is it? It involves eliminating processed foods, sugar and other potential foods that trigger consuming a diet that is rich in whole foods, fiber, probiotics and prebiotics. The aim of the program is to reduce inflammation and promote the growth of beneficial gut microbiome. Now, what should I say? Nowadays, the inflammatory, it's very easy to note the inflammation because interleukin-6 has become available everywhere. So any chance of inflammation can be found out, especially after the COVID event. So we can look for interleukin-6 and find out if there has been any inflammation of the gut and any dysbiosis. Now the effect of fermented foods, we know Indian foods, though we are talking about kimchi, etc., which are not Indian, but we know we use a lot of fermented foods. They have been used for time immemorial, for thousands of years, and these act as probiotics and supply essential nutrients. And definitely, if you have to change a diet, go in for fermented foods. As a microbiologist, I'm ready to hold your hand because dietitians and microbiologists go together at this point. So, effect of the fermented foods. So, what happens is you're supplying a bacteria or fungus-laden food which has already been broken down into simple nutrients and which will be good for the patient and will also diminish the inflammation of the gut. So benefit of the fermented food, we can see an overview. So there are health benefits, it changes the gut microbiome and it can modulate the gut microbiome. It increases the gut microbial diversity. A Stanford study showed that a 10-week diet high in fermented food like yogurt, kimchi, etc. led to a significant increase in microbial density. The diversity is associated with improved health outcomes, including better immune responses and reduced inflammation. So, reduction in inflammatory markers. So, we all know that there are 19 inflammatory proteins, including interleukin 6, which we always look for as an inflammatory marker. Now, regarding the probiotic content, I've already talked about it, the metabolic benefits. There are high levels of, just like my previous speaker has said, it supplies conjugated linoleic acid, CLA, a molecule with potent health benefits. So it replaces the gut microbiome. Now the prebiotics, we all know what are prebiotics, and the use is to restore microbial balance by stimulating the immune system and inhibiting pro-inflammatory cytokines. What are the main probiotic families used? The lactobacillus, the bifidobacterium, the lactococcus. What are prebiotics? It is essentially the fibers that are undigested in the upper part of your GIT and it goes to your GIT so that your microbiome can feed on it. What are synbiotics? Probiotics plus prebiotics make synbiotics. What are pharmabiotics? You add some vitamins, make them more genetically stable, it turns into pharmabiotics. So it can deal with irritable bowel syndrome, chronic idiopathic constipation, and abdominal bloating. I've given you a list of organisms that can do it. So if we can grow them in the laboratory and we can take patents and then perhaps we'll sell it to you and your patients and we'll become very rich. Come on, let's see. And then FMT, fecal microbiota transplant. Regarding this, even the last issue of Nature said, this will never be a popular, this diagram is also, this photo is also from the article that Nature had given. They have said in the article that even now it will be a very difficult concept to propagate. So it can act against primarily it was for Clostridium difficile infections, but it can also be used against, I think I've got very little time, so I should not get into the procedure or anything, but it has a potential to act again, inflammatory bowel disease, irritable bowel syndrome, metabolic syndrome, neurological and psychological disorders. And in a number of them, FMT has been tried and tested and it is successful. But what happens is, if you don't do it in your own hospital, actually you'll never know what gut flora lives in our part of the country, what is present in the people who live in our country, and what is the one that makes people intelligent or resists the different disorders like autism, autistic spectrum diseases, depression, Parkinson's disease, etc. I'm skipping the steps, but one thing should be said here, that a patient for FMT should be chosen whenever there is no neutropenia. Two things should be ruled out. One is there should be no neutropenia, and the other is there should be no inflammation of the gut. So these two are very important things that should be kept in mind. FMT holds a great promise beyond CDI, and ongoing research will help to clarify its role. Now regarding fudge therapy, this is also from Nature. So have you ever seen an African safari? What happens there? There are lions running behind herds of gazelles or deer. And then what happens is, the one which is the weakest or the one which is sick, that is killed by the lion. So exactly the same thing happens in our gut. The gut bacteria are there and so are there the bacteriophages. the gut, the bacteriophages attack only the bacteria that are sick, that are weak, and in this way, and that are less in number. So whenever a pathogen gets into the gut, what happens is, since it is less in number in the beginning, it can be killed by the gut bacteria. So it regulates population, there is horizontal gene transfer, there is impact on the health and disease. Now, it gives diversity to the bacterial population and it can do immunomodulation. In a number of cases we have seen, phages have the immunomodulatory role. So it is not only the bacteria but also the bacteriophages. Do you know what are bacteriophages? The viruses that feed on the bacteria. So I had been talking all about phages and I did not say what were bacteriophages. That was a very big mistake that I made. So they act as predators on bacteria. So we can engineer them in such a way so that we can kill the pathogenic bacteria. And we can also use it for immunomodulation. So what are the challenges? Complexity of interactions, methodological limitations, and need for more research. See, one thing about it is the gut microbiota needs help from everyone. Every aspect of the faculty should come forward and they should help us to chart down the actions and the composition of the gut microbiota. And last but not the least, it's microbiome engineering, directly manipulating the gut microbiome through genetic engineering and synthetic biology that will be coming in very soon. So we all know that CRISPR Cas systems. So that is very important here. We cut the gene, place it somewhere, we make the ideal phages, we make the ideal bacteria that can help to improve the microbial genome. Applications is, for example, engineered probiotics can be designed to produce therapeutic compounds or to outcompete pathogenic bacteria. So here, nature had said about a very difficult situation where in Switzerland, where they make cheese, they had supplied bacteriophage to the cheese makers. You know, you need bacteria to make cheese. So first time it was supplied to them, what happened was all the bacteria died. And then the people, the scientists, they were at a loss. So what happens if my phages kill all the bacteria? That is what exactly happens in the gut. The phages kill the good bacteria. So what happens is you have to find out bacteria that are phage resistant, use them to make the cheese and other probiotic food. And then if you use it for your patients, in that case, the patient will survive and definitely you will have better results. So, thank you everyone. Here I can show you it's a phage, lot of phages feeding on the bacteria. This is also from nature. A transmission electron microscope shows bacteriophages attacking a bacterial cell. Thank you everyone for your patience. Actually, this topic is not so much related with you, but we need collaboration, and that is what I need to tell you all. Thank you, everyone.
Thank you so